Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out from my PhD in microbiology. Then I was a fact checker or editor for pharmaceutical advertisers. And nowadays, I'm the president and a member of Biotech Without Borders, a nonprofit based at NYC, dedicating the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked in bacteria, but I've also worked as a research integrity specialist, and I'm currently working for a scientific journal.
Every week we meet to talk about microbiology, and on a typical day like today, although we've been away for 4 weeks, we do an overview of some of the coolest microbiology papers that we've seen in the last 2 weeks.
And at some point, we hope to focus in on one of these papers to do a deep dive or do a deep dive on the topic generally. So if you have any suggestions for things you want to cover, please make sure to message us at microtwjc. Let us know if something's caught your fancy.
And you can follow along with any of the papers that we discuss in our shared Zotero library, linked in the doobly-doo below. And our social media handles are in the link, and there's also, a link to our Zotero library down there.
And we want to hear from you, so please please like, subscribe, and please use the comments to tweet us, or tweet us at microtwjc. And boy, do we have a show for you today. First up, we have some updates on monkeypox outbreaks, a note on how we can speed up SARS-CoV-2 testing before we dive into the meat of the show, which is what's next for SARS-CoV-2 vaccines, and what do we need to do to make them better. But not only that, we look at Mpro, the target of Paxlovid, and whether there are SARS-CoV-2 strains out there that are resistant to it, and if not, will there be? And then we have type 6 secretion systems, also known as molecular crossbows.
I was wondering about that.
How bacteria use them to fight for niche space in your gut. And finally, we have RNA wrestling for space on a carrier protein called HFQ. And what does that mean for cell signaling? So stay tuned. So jam-packed show.
First up, we have this monkeypox paper that you picked out. Monkeypox virus infection in humans across 16 countries, April to June 2022.
Yeah, that's right. So this is mostly just a reminder that monkeypox is out there, and it is I think the technical term you looked up just a minute ago was a global public health emergency, right?
Yes, a global public health emergency. That's what the WHO has indicated it as.
If only there was a single word that we could use to encompass that. If only there was a single simple word, maybe just for fun, it begins with a P, but who knows?
But I think as we spoke about in the last time we talked about monkeypox, like the course of infection is a much slower course of infection than what we saw with SARS-CoV-2. So the thought of I guess the speed at which we see it move around the world, it's actually a little bit slower, but also harder to detect in some ways, because in terms of sort of the speed at which people become positive or seek testing. I think we also had said something in the past episode doctors might not know to order the monkeypox test, but part of the raising the alarm about it is to inform people that this is something that should be tested for if certain symptoms align with the presentation of this virus.
Yeah, I think this paper does highlight that specifically, because I think they mentioned that I think because in this study they go and test a whole bunch of people across different countries, and I think that quite a large proportion of them, I think, tested with only a single genital ulcer, which is small, which could easily be misdiagnosed as a different SDI. So if you just have one little bump or one little kind of nasty spot, that could be monkeypox, and people might not think that it is that, because a lot of what we hear about it is for a much more distributed rash and other things. So there is some diversity in how it presents.
Yep. And this is a clinical paper, so it comes with the requisite horrifying pictures of the symptoms.
Yes, it does.
So for those that are interested, these are some of the presentations. And actually some of the pictures it's not so much sometimes it's just within the mouth, it seems rather than actually on exposed spaces.
Yeah.
Could be
so yeah, they go across they test they found 528 cases that are all PCR confirmed, and they kind of catalog the cases where they're found, and like the majority of areas that are affected were the skin, specifically the anal genital region, but then you've got like some on the nose and throat. You've got some in the trunk and limbs. So it's because this is a skin disease mostly, like a cutaneous disease, and it just happens to be affecting certain areas of the body in this case. And part of that is due to its impact on the MSM community, the male sleeping with male community. So that counts like bisexuals, gay men, and various other kind of iterations of those. But essentially, it's still a big thing. And it's very much a slow motion pandemic that it's now entered into like the international like network of travel that happens in quite a lot of Western countries.
Yeah. I guess that's all I really am interested in about this. I've gotten kind of curious about, because at least in the US, I've heard in many countries, right, they're rolling out vaccination strategies for this. And so, I'm a little bit interested in that, but we don't have any information this week for you guys about that.
This is mostly to keep this on your radar because it's certainly on ours. And now it's going to move on to the next paper, which is the next big glamorous virus outbreak that is still going on.
This one's avoiding false positive SARS-CoV-2 rapid antigen test results with point-of-care molecular testing on residual test buffer. And this is like I guess like a more of a like strange technical testing paper that came out, and I wanted to just bring it to people's attention in case there were any questions, I guess, about the rapid antigen tests and like how they work sort of with PCR or like how they're different from PCR. And maybe people have already sort of thought about this, but you know, those rapid antigen tests, like you get a little swab and they're looking for some proteins inside of the swab that will like give you that positive band. But of course, like because of where it's got swabbed, it could have all sorts of material from the virus. And so in this paper, they take that buffer that you usually drip into the lateral flow chamber or whatever, dab into some sort of moisture thing, right? That brings it through capillary action through the test strip. They take some of that liquid and they sequence it, or they put it through. I think it's actually it's like this. It's like a machine that does PCR to try to figure out if the sample is positive or not. And they're in some ways double checking this one test modality with another test modality, and then they provide some commentary on that. So there seems like it works pretty well. I think we've said it before too. Like rapid antigen tests give you a good indication. Like all of the ones that get tested that come up positive, they're also positive by this PCR. I think there are a few folks that like maybe can come up positive with PCR that don't come up positive with the rapid antigen test, but those also are not thought to be so infectious. They don't discuss that in this particular paper, but they sort of refer to some other ones.
So this, I imagine, is going to come in useful for people who are like traveling a lot or have to like do tests quite quickly trying to get people out of quarantine as quickly as possible, basically. So that when you have a false positive, that's a really bad thing if you're trapped in quarantine and you don't actually have coronavirus, but having that positive test, that's a bad thing. So having anything to confirm that, or like that, can actually save a lot of people's time. And so having something like this where they have this thing where if you test positive, that isn't the end of the story within like hours. So they've got this portable rapid diagnostic test, so to test for nucleic acid. So which happens, I think, within a couple of hours. So even though you have that test and it comes up positive, there's always a chance they can do this extra test just to see, and then that comes back negative, then you're free. You're out of quarantine. So I think
so
from a practical perspective,
it's interesting too, because I'm not sure how many places still have testing requirements around them. Like I know that at least between my parents came to visit recently from Canada, and like there's no testing requirement across that border anymore, like Canada to the US. So yeah, I guess in some ways maybe some of this information is like people aren't using it anymore because things are just back to business as usual. But for places that aren't, or if people are trying to think about like where tests should fit into their their strategy for crossing around or like reducing risk within their own networks, like this might be a useful thing to think about.
Yeah, I mean, whereas I'm going to be going to Germany, which again has, I mean, places in Europe still have it's very confusing what kind of testing regimens there are out there. So have to make some way to get tests done. So yeah, it's.
So what do you have to what sort of tests do you have to do to get to Germany?
I don't know yet.
Do you know?
It's very unclear.
Okay.
Because what different places say are quite different, so it's all very confusing. Part of it is because of this thing called Brexit we have in the UK that has made all sorts of international communication quite difficult and confusing. So on to the next paper.
Okay. Oh, this one's kind of cool. Or this is a weird one. I didn't expect vaccine information to come from bacteriophages, but here we have a bacteriophage-based, highly efficacious, needle and adjuvant-free mucosal COVID-19 vaccine.
Yeah, this is the first paper looking at we'll start with the big one, the one that has quite a lot of bells and whistles on it. So firstly, we've got bacteriophage bases. Bacteriophage, these tiny little viruses that feed on bacteria that don't generally interact with humans very much.
Yeah, I feel like we've talked about it on the show in the context a lot of, well, like weird ecological interactions, because there's so many bacteriophage in the world. They do lots of funny ecology things with other bacteria or even other bacteriophage. Specifically, when you think about sort of like natural selection for genes that they want, there's like different weird arms races that go on that can get very complicated. We talked about that in previous. And I think we've also talked about bacteriophages in the context of phage therapy, right? Using viruses against bacteria, as a therapeutic of some sort. But this is bacteriophage for us, for our immune system.
Bacteriophage against a virus, a human virus, because I think we covered this before. Bacteriophages usually fuck with other bacteriophages. They usually like mess with other viruses that feed on bacteria, but not with viruses that feed on humans. So this is again, so trying to pass it together. What is going on here?
But at the end of the day, like it's not that the bacteriophage that they made in this paper is like specifically targeting, right? It doesn't have like interactions with the virus that we're making. It's almost like a manufacturing platform that they just want to make a particle that has interesting immunogenic properties towards COVID-19, but they're going to make that particle using bacteriophage. So the bacteriophage, like just like any virus that has like this little, or many viruses have a protein capsid, and so this bacteriophage is no different. The investigators in this paper engineer that capsid to have the S protein, or the spike protein on the outside of it, of that surface. But then, and inside of the capsid, they have it make nucleocapsid protein from the SARS-CoV-2. So it's kind of like, I mean, in my mind, it's almost like it is like a nanoparticle production system. The bacteriophages are, we know they have a certain molecular configuration, and if we substitute in certain proteins of theirs with the protein that we're interested in immunizing against, then we create like a kind of a cool structure that can perhaps drive different types of immune responses than making it ourselves or in some of the other modalities that vaccines get made.
Yeah, I mean, there are a couple of like different, I mean, different things that apply to that. So firstly, the proteins they select to show. So I think we use this CRISPR system to generate multiple vaccine candidates. I think I should probably talk about why this is important. What we already have SARS-CoV-2 vaccines, so why are we wasting some? Why are we developing more? Well, firstly, because the SARS-CoV-2 vaccines they don't prevent transmission, which is something that's very important. And also, the variants. There's so many new variants coming out that are evading the vaccine. So having a broader target that can prevent evolution away from it. So this addresses a bit of all of them, actually. So firstly, we want to make sure that that variants can't evolve, or that variants are very difficult to evolve. So what they have here, they've got multiple proteins, as Danny mentioned earlier, that they've got capsid, they've got like a spike protein, they've got various other like nucleocapsid proteins in there.
Yeah.
And
E
more,
protein was also in here.
yeah.
Yeah.
So they're producing this wide like batch of proteins to make sure that SARS-CoV-2, in order to evolve away from this vaccine candidate, it has to do multiple strategies. It can't just get away by making a few mutations on the spike. It has to mutate some very key proteins. And so the idea is to develop this broad immune response that can immunize the original strain, Beta and Delta, and hopefully Omicron. And then the second thing they want to do is to make sure it doesn't transmit. So that is where the other aspect of this paper, the interaction with the
Needle-free.
mucosa.
Yeah.
So towards that, the design platform. So they're showing all these proteins, but these proteins themselves are almost independent vaccine candidates they've screened to make sure that they're going to be as immunogenic as we'd want. Yeah, they've got so much stuff here.
Yeah, so they test it both intramuscularly, like injecting it into the muscle tissue, and also intranasally, which is just like instilling it in the nostrils of these mice. And they get fairly good responses from both, but in that intranasal route, they get the mucosal response, which is like antibodies being made on the mucosal surfaces. I'm not sure if we've talked that much about it, but IgA is like the antibody that's made in that circumstance. And that addresses this issue that that addresses the issue of transmission a little bit more than other modalities of giving vaccines, or other modalities of immunity rather, because like that's the surface that we shed the viruses from, or first come into contact with those viruses. So having like a protective or some sort of immune response in that layer could prevent transmission a little bit better. I don't think they don't test like actual transmission in the model though.
No, they don't, but they do they do test to see whether there's an IgA-like immune response happening, which is quite important. Let's see what else do they do.
I was going to say another nice thing about having a vaccine like this is I think it's not going to need the -80 for being stored. That's one of the thoughts. Yeah, so it's a more stable. It's like the these molecules are just more stable.
It's a protein vaccine, so like the other protein vaccines, it doesn't need as much. Proteins are a lot more like a lot less delicate than things like RNA, so they can be much more stable. And especially since they're in virus form, and these like bacteriophages are designed to be pretty stable because they have to exist in the environment. Another interesting thing is the idea of these vaccines are going to be designed to be more inhaled, again, which is an interesting thing because I think part of the thing that people hate about vaccines is the needle part, and having something you inhale is a lot better.
Yeah, well, maybe easier for people to take, requires less training to administer. I think that could be one of the advantages. Although I'm pretty sure you still want like a healthcare professional to administer the intranasal vaccines. I've gotten intranasal flu vaccines in the past.
Right. Yeah, I'm somewhat skeptical about this because I think the method by which you make something intranasal for a mouse might be quite different for a human, because the way that my mouse's nostrils are set up are somewhat different. I mean, because I know I work with an intranasal vaccine candidate, and what essentially we do is we kind of we don't kind of spray in their faces. I think it's just literally putting in a drop of water onto their nostrils and letting them inhale it in. Which
installation,
is pretty uncomfortable if you ever had to do it as a person.
Right.
I've never done it as a mouse, just to clarify. I don't know why I phrased it like that.
Yeah, I mean, there's work that has to be done, right, to adapt what is the intranasal administration in a mouse to intranasal in a human, and then who knows, right, if some of the effect goes away in that adaptation process.
Yeah, and the question I have in my mind is okay, so this is a protein vaccine that shows a wide variety of vaccine candidates, but how is that different from the inactivated SARS-CoV-2 vaccines, which haven't proven to be as we'd like? Because essentially it's almost presenting a similar thing. I think the main differences are that it's the targets themselves have been screened, and they're in their original conformation. Also, there's this attachment to the mucosa, so they've got these immunoglobulin-like fibers that like stick out from the virus that allow it to stick to the mucosa and might be immunogenic in themselves. It might, or just by itself, annoy the immune system enough to make it form a response.
Yeah. I mean, it's the viral particle as the adjuvant, right? In some ways,
Yeah.
it's like directing. It's both. Yeah, it's sort of modulating the immune response in a certain way. Yeah, I thought that is interesting actually. Like, because these are the proteins of the virus, the T4. I think they're the Hock domain.
Yeah.
Yeah, like they're part of T4, the virus. So like, I wonder why this virus does, right? Like, why does it get attracted to this particular area? Or is it just also because everything sticks to mucus, right? Like the highly repetitive polysaccharides of the mucus is like just a thing that many things stick to, and it just so happens that this virus has some stuff that sticks to it as well.
And another thing that I was wondering is, like, since it's a bacteriophage, can you get it to replicate in the natural bacteria of the human body? They don't really, they're not aiming to do that in this study. Right. I think they take some care to say that it doesn't affect the microbiome community.
Yeah, that's almost like one of their preliminary like safety checks inside the model, right? It's like they don't want to say, because you could imagine like, oh, I'm putting way more of this phage inside of my body. Maybe I'm going to have this crazy dysbiosis. But yeah, they show that it doesn't.
Yeah, I think that's everything that I wanted to really pick up on about this paper.
Yeah, I think a really cool, a really interesting vaccine modality. And I do just briefly want to say, I don't think we really talked about it, but Novavax got approved at some point, right during yeah, finally. And like that was the sort of the forefront runner of protein-based vaccines. And again, like it's a really great thing because it doesn't need the same storage requirements. Presumably, there's different types of like manufacturing facilities that can make that particular vaccine. So it's really good for the global vaccine effort, which is not right now. And we touched on it just before. Like we want to have a good global vaccine effort, whether it's from Novavax or other emerging vaccine candidates, because that reduces the amount of flexibility the virus has to evolve within our population and find, some other way to cause chaos.
Yeah, because I mean, viruses don't see borders, and if your virus response like stops at your own border, then you can't control what new variants can evolve in other places, and those variants can always travel. So you have to think, we have to start thinking about health globally, especially with terms of like viruses and diseases.
Yeah. Which is kind of a segue into the next paper, because like one of the strategies I think that has emerged in this era of viruses everywhere, like, COVID everywhere sort of thing, is that we still need even better COVID vaccines because of the emergence of variants, and figuring out what makes that better COVID vaccine. How do you make a vaccine that will hit the variants is something that is on some scientists' minds.
Yeah. So the title of this paper is Structure-Selected RBM Immunogens Prime Polyclonal Memory Responses that Neutralize SARS-CoV-2 Variants of Concern. So yeah, this is an interesting one because the receptor binding motif is the part of the spike protein that binds onto ACE2. And I think what this is trying to look at is they're trying to target that interaction between ACE2 and SARS-CoV-2. So the current vaccines only target the RBM or like areas around that, basically. Whereas if you target both, then you're literally targeting that specific interaction, which might have an interesting effect. So they look.
Yeah,
sorry.
I was going to say, like, one of the terms that came up in this paper that I didn't know before is paratope.
Yeah.
Where paratope is like where the antigen gets bound to in our immune system, and epitope is that antigen that is binding to that section. So like part of the research in this paper is, I guess, making a molecule that looks like our T cell receptor that has like a particular epitope already put inside of one of those T cell receptor loops. And that's being used to stimulate B cells.
Yeah. So let me see if.
But okay.
Because this is an interesting one, because like what they do is they take this kind of sequence and they inject it directly into the spleen.
Yeah.
Actually, it's even more crazy than that. It's almost like because this is almost like a gene therapy paper that is masquerading as a vaccine paper, because they've got this specific like paratope, that specific like receptor that binds to this, and they have like it put into a vector. They put into the spleen, and the idea is that this will go into the spleen, enter into the genomes of the B cells, and kind of skip that step of recombination, all that decision making that the body usually does in order to create the right receptor. I think I want to double check this because I need to make sure this is the right paper.
Yeah, it is something like that. I'm not sure whether or not this construct goes into B cells or T cells. My thought was that it went, again, this is just from my quick skim over, but I thought it goes into T cells, and those T cells somehow interact with B cells. But irregardless, the idea is sort of similar. It's not about providing like the thing that the foreign object that the immune cell is going to respond to. It's sort of providing what you think the immune cells should look like into the spleen, specifically into the spleen of these animals, to then so that process of selecting the best cell, right? Like those cells are already, you put the cells there, like you put the best cell, so to speak, into that population, and that gets amplified within the system of the animal.
Yeah, and the spleen is the organ that does a lot of cleaning up of the blood, and it is filled with immune cells. And here and yeah, this is just a fascinating way to vaccinate, because they've got a target in mind, and they're taking away any of the body's decision making in this. It's saying that we know what we want to target, so let's just put that directly into the spleen, transform some B cells with a specific receptor, and then go out there the way we think that they should do.
Yeah. So it is B cells.
I believe so. At least that's what I wrote in my notes when I was reading this paper. So
nice. Okay.
This might have been a problem for me a month ago.
Yeah. So kind of, I guess it shows, one of the things that I think it shows is like our understanding of what the epitopes look like, or what the opposite response, right? What the paratope looks like inside of a mouse that corresponds to the right type of immunity that we want. And so that could give us better control. I don't know how this would go into humans, I guess. Maybe it's like really not about that bridge into human vaccines, but it's like more the knowledge of like what epitope corresponds to that is useful. Yeah.
I think so. I think this is very much a toy model to test an idea, and they couldn't trust the immune system of the mouse to create this kind of paratope, because I think there's a lot of training in the immune system to make sure it doesn't hit the self. It doesn't hit like the things that are seen within the body. So maybe that'd be, it depends on whether they're using it, because again, human ACE2, so maybe that isn't it. So
well, no, I think they're trying to be specific about directing a response to like something really, like, because if they use like a larger piece, I guess the question is, why do it this way but not just use the smaller piece of this specific region, right, to direct the response? I think they even said something about that though in the intro, and I just don't quite remember. It's all before figure 1 would be these planning steps.
For me, I think that they didn't want to leave up the chance. They had a certain effect that they wanted to see, and they didn't want to leave up the chance that the immune system might create a completely different antibody that wouldn't kind of be honest to the experiment they wanted to look for.
Right. Right.
And so they did test this on a variety of different strains, and it's not like a perfect response. So they had, I think, a set number of different structures that they tested. So I think they said like 1, 2, 3, and 4, and some of them worked better for like say the beta and the delta, and some of them might have worked slightly better for like Omicron. And so I think that this in itself demonstrates that maybe it's not the most consistent of antibodies. Might not be the most like
or even directing an antibody towards this specific RBM region isn't, like there's still some variance between the there's still some differences between the variants that might make that effective at different varying levels.
Yeah, so you've got like, so for instance, they had one type of I can't remember, I think one type of epitope that they used, and type 1 seems to work consistently across all of them. But then if you look at 3 and 4, that's a bit more variable. 2, that also is a bit more variable. So I think that from this, they're kind of learning a bit more about how the immune system can react. I mean, moving towards that idea that maybe we can in the future be able to design better immune responses than the immune system itself. So I think that's interesting, but I think mostly the testing this kind of, I mean, yeah, testing whether this kind of targeting can work.
Yeah. Okay, yeah, I think that's all I want to say about this one too. So the next paper that we have is, again, trying to figure out, again, it's all about targeting antibodies to different places. And in this one, they're using S2 targeted vaccination. And again, elicits broadly neutralizing antibodies is the title here.
Yeah. SARS-CoV-2 S2 targeted vaccination elicits broadly neutralizing antibodies. And this also picks up from a bit of research that we heard very early on during the outbreak, but didn't. So I don't know if you remember, there were like some cases where people had less severe SARS-CoV-2 because they were infected prior with another coronavirus, OC43.
That's like the human circulating common cold style coronavirus. Yeah.
Yeah, there was.
Oh, I remember there was something where they found antibodies towards regular coronavirus in people that hadn't been previously infected at all, and they traced it back to this particular like circulating variant.
Yeah. So in this paper, they kind of follow up on that by looking at the S2 proportion of the spike protein. So for those who don't know, spike protein, there's an S1 proportion and S2 that get cleaved during it. And the ones that we tend to be focused on are the S1, because that's where all the grippy parts of the spike protein are. And then the S2 is mostly where the fusion bits are located.
Right. That cleavage is the furin cleavage, right?
Yeah, that's right.
And then there's also like, yeah, and so like we've talked also that like different variants like have different sensitivities sometimes to furin, or like maybe autocatalytic, maybe use other proteases. Yeah, this is like part of the flexibility of what we've seen and the diversity of different spikes.
Yeah, and what they kind of found was that the S2 seems to be quite conserved across a lot of, not just SARS-CoV-2 variants, but also across like coronaviruses themselves. So they kind of wanted to, so invasion by the immunity by the VOCs, the variant of concern, your Omicrons, your Deltas, that tends to be linked to the S1. So they're lightning focused on the S2 unit. So what they kind of did was they looked at what happened if they use S2 as an S2 based vaccine, and seeing whether they could protect people with, like, say, so for the first pet experiment, they use an OC43 primed mouse, almost like mimicking that, and then they tested to see whether the serum from that could react to SARS-CoV-2, or and I think it could, but only the S2 portion, and maybe a little bit of response. But no, I think it's just for the S2 portion. And they do a couple of other kind of studies on this to essentially find that if you imprint specifically with the spike protein, the S2 portion, that produces a broad coverage of immunity that can cover quite a wide variety of strains and species, which is good. The only kind of not quite as immunogenic. So it produces an antibody response, but maybe not as high as a response as you'd want. And they compare this to just giving the full length spike protein. And if you have the full length spike protein, the immune response almost naturally goes to the S1 proportion rather than the S2. So you have to directly tell the immune system, you have to imprint the immune system with an S2 response in order to make sure it will respond to that when you expose it to the real SARS-CoV-2.
Yeah. Yeah, so it's, we're trying to direct, back in that line of thought from the last paper, like, after we've looked at sequences, right, of the virus, we're saying, well, it would be really nice if we could have immune response to this more conserved region. in some ways, we're thinking almost about universal coronavirus vaccine sort of style going on here.
Yeah.
And yeah, and that could be incredibly helpful because like that's not where the majority of the variation occurs between coronaviruses, which makes this vaccine more robust against a lot of different evolution that could be happening.
Yeah. I mean,
Yeah.
the interesting thing also is that the immune imprinting effect is so important in this that they that if they think they try to replicate to see what happens if they've already vaccinated using the kind of vaccines we're already getting, and seeing what happens if they did a boost of the S2, and I think it didn't work as well because of that previous immune imprinting. So it's kind of fascinating, and this be interesting for.
Well, that particular finding sounds not very useful or like for the way things are, right? Because most people or many people have been vaccinated against the first style. So it's like if you can't shift it, then then this strategy isn't as useful. I'm also thinking about how they're saying that the next round of vaccines might be Omicron specific, but that's most undoubtedly Omicron specific in the S1 and receptor binding domain regions.
Yeah, I mean, you're right. It's not going to be useful for me or you, but say the next generation, say like the next generation of kids, it could be quite useful for people who haven't had that immune imprinting yet. So it's something to think about for like further along the line.
Yeah.
Especially.
Or I guess, I mean, there's still the opportunity in unvaccinated populations, right? That this could be a useful vaccine strategy.
Yeah. I mean, if you're unvaccinated and have made this far without, say, getting SARS-CoV-2, then it could. And I
get vaccinated. No, get vaccinated.
Yeah.
Sorry, still get vaccinated.
Yeah, I'm sorry. I'm not going to tell you to wait for this experimental vaccine. It might never come through.
Yeah, but yeah, I know it's interesting to think of how this vaccine might play upon us. But maybe again, it's like, as a lot of things that we've seen, especially around like the emerging technologies, using SARS-CoV-2 almost as just a model to understand the way our immune system works, then maybe here we're learning a little bit about just coronaviruses in general and strategies for vaccinating against them. Yeah, even though maybe it's compelling to think that, wow, like this could be used with SARS-CoV-2, it's a little bit more complicated than that, just given how many people have already been vaccinated / have had an infection. I think that might be something. If I was trying to read this paper a bit closer, that's something I might dive into a little, right? Like, what about just previous infection from some SARS-CoV-2 strain, right? And then giving the boost against S2. Can you push towards the S2 through that mechanism?
Even like the human coronavirus as well. I mean, we test using different coronaviruses to see how well the imprinting actually crosses over between species. Because I think like even if we can't like vaccinate against SARS-CoV-2, having in mind that there are other coronaviruses out there, this could be useful, a useful stepping stone, especially if one day we find a way to solve the immune imprinting problem. But yeah, that's why I found this one quite interesting.
Cool. All right, so the next paper we have on the docket is Genetic Surveillance of SARS-CoV-2 Mpro reveals high sequence and structural conservation prior to the introduction of protease inhibitor Paxlovid. So essentially this is talking about setting up a surveillance pipeline to look at Mpro. So, you know, there's many tools that we have against SARS-CoV-2, which is actually really great. I think that's part of why people have been so enthusiastic to just go back to quote unquote normal. But of course, like that's like things, you know, multiple barriers are still really useful for public health, and a concern could be that the introduction of a drug could also produce some sort of resistance. And this paper actually is specifically looking at, was there resistance in SARS-CoV-2 diversity before the introduction of Paxlovid, and that might create a baseline for us to understand as we continue to monitor the evolution of SARS-CoV-2, and specifically now paying attention to Mpro, will resistance emerge as this drug is being used more and more to treat sort of certain patients that have SARS-CoV-2. So yeah, that's this paper. So Mpro, just the background for Mpro, that's a protease that's being made, one of the first proteins that's being made from SARS-CoV-2. It'll chop up, I think, the polymerase, the first, right, the RNA-dependent polymerase so it's like, this protein's important in the very early stages of getting infected, of having a cell get infected by SARS-CoV-2. It's highly conserved across multiple coronaviruses. Sorry, go ahead, Fuzz.
So yeah, no, I was just gonna make an analogy. So it's like, you know, the Airfix model sets you used to get as a kid. I don't know if you had this, but where you have these like kind of pieces clicked together all in one kind of board, and you have to click them out. This is a protein that does that.
Like making models? Is that what we're talking about?
Like making models, yeah. They always come in this like linked plastic sheet, and you have to like kind of pull out each bit. This is the protein that does that for SARS.
I like that, and yeah, we can link that back to them being very viruses being very space efficient with their proteins, right? So they just have like the first thing they make is just like everything fused together with all that plastic flash and extraneous things, and then they have a different protein that comes along and pops out the necessary ones so that they can go on and do their things in the cell.
So yeah, when Paxlovid comes along and binds to like the area where it binds, it stops this protein from activating. So and this protein kind of needs to dimerize in order to, so almost like as in terms of like a set of scissors in terms of like how it kind of pushes things out. So I think the big takeaway from this paper is that they looked around for kind of things that could potentially be inactive or kind of make the binding site inhospitable for Paxlovid, and they didn't really find anything. I think. They're like,
which is good because it means that in the existing variation, the existing variation of SARS-CoV-2, like there isn't anything that could be selected for out of that population that then would be resistant to Paxlovid. I think that's very promising.
Yeah. So this is great for.
Doesn't mean that it couldn't emerge, right? Like because like variation may constantly be generated, and of course like the longer the pandemic rages on, the more chance of those things exist. But in the current sequences that they have in their database, nothing's poised to sort of overcome this particular drug.
Funny you should say that, because that brings us to the next paper. A comprehensive fitness landscape of SARS-CoV-2 Mpros reveals insights into viral resistance mechanisms. So this is a paper that doesn't look at what's out there. It looks at what theoretically could potentially happen. So in this case, they use this thing called systemic mutational scanning to assess the consequences of all point mutations in the gene. So what they're doing is like they're changing every single piece of the genome of this like kind of enzyme. Genome is the wrong kind of term, but each part of the DNA sequence of this enzyme to see whether there's any iterations that could make it resistant to Paxlovid.
And they do that in yeast, and they have like a whole bunch of different assays where, you know, they have some molecule that if it gets split up, it's going to glow, or like a molecule that if it does get split up, then it doesn't glow. And I think also Mpro, it can also kill yeast cells. So they have like a bunch of different assays that they use to try to get at, basically locate those sequences that is inactivating, the ability for inhibitors to work.
Yeah, and they did a bunch of mutations, and it's interesting to look, because if you read deep into this paper and look at the last paper and look at what things are the same and what difference, it is actually quite interesting to see like which ones they find are very mutation sensitive, which ones like can the Mpro like not function. I think the important thing is like they want to make sure that the testing which mutations make this Mpro completely non-functional. And then which one, so showing what's important for the protein, which the other paper does a little bit of as well.
Yeah, but I guess like I like this one. Yeah, the other one does it in terms of like what's out there that isn't possible. This is very much like what's theoretically impossible, right? Like if you make certain mutations to Mpro, like it's just not going to work. And this goes back to we can even say that this sort of thought process goes back to thinking about like the SARS-CoV-2 spike protein evolving away from things, right? Like at some level, if the evolution acts on the spike protein or Mpro and pushes it to where it's non-functional anymore, then that's not going to happen because like it means that infection won't be productive and we won't see anything. So there's some like Venn diagram of mutations that we can see, right? That that preserve the function, but then disrupt the ability for the intervention to work on it. And so in this case, it's about binding to the small molecule inhibitor. the Paxlovid,
and they do like some actual studies to kind of check what kind of mutations can allow like Paxlovid to not bind, but also still keep its for the protein to still keep its function. And they find, I think, 3 mutations that have potential resistance.
So in some ways, it'd be kind of a nice. It's like these 2 papers really do feed off of each other because like this paper, if the last paper, the primary contribution, besides like the insights into Mpro, is also like the pipeline of observation, like watching out for where mutations are emerging, like because they did all this data processing to get there. This paper can provide some insight into what sites are interesting to watch. Like if we end up seeing variants that pick up one of these mutations, we can start asking the question like, why did it pick up that mutation? Is it because like Paxlovid's being used in some locality and we're seeing evolution, right? Like there's still enough spread that it wasn't being suppressed properly.
Yeah, so these kind of paint the picture that there isn't any resistance to Paxlovid at the moment, but it is possible, and we now have ways to kind of detect those resistant mutants when they appear.
Yeah, we know what to look for. We know what the trouble sign would look like, but it's not out there yet.
No, it's not.
And hopefully it won't be, right? Like, and I think critical to that hope is like this collective effort to reduce infections.
Yeah, which brings us to the end of the SARS-CoV-2 portion, and now we're on to bacteria. So type 6 secretion systems of pathogenic and commensal bacteria mediate niche occupancy in the gut.
So story of the microbiome, and specifically the microbiome inside of a model, a diarrhea model in mice, where you feed them some of this Citrobacter rodentium, and the mice get diarrhea, but not always. and so this paper is investigating what that not always is. Actually, they already have a framework for thinking about why this could be the case, and it's the molecular crossbows, as you said in our thumbnail, right? A type 6 secretion system, which injects proteins from one bacteria into another bacteria, and that mediates a lot of competition between things in our gut. And it's thought that basically the different types of type 6 secretion systems and the different effectors, the bolts that they have loaded into them, really give us the susceptibility or the resistance that our gut has to different other transient organisms that are passing through.
Yeah, one thing I have to say is that this paper is the best description of a type 6 secretion system that I've ever read, because they describe it as a molecular crossbow, and they describe each kind of like piece almost as like, oh, this part is a spring, this part is the thing that delivers it. It's like, and they describe like proteins as part of the needle and the like kind of the tip of it, and how that interacts. So it's
that's great. That's funny. I thought you dreamed that molecular crossbow up yourself, but it came from this.
I did not. I would have done something more esoteric. That I have a thing of like making analogies of things that are even less popular or knowable than what I'm describing.
I guess like I certainly skimmed over then that analogy in this paper, because like in my mind, or like the thing I always think about with type 6 secretion is it's like similar to some phage stuff going on, right? Like the phage injection systems, it has some relation to. But anyways, the Citrobacter rodentium that they're using, it has 2 different type 6 secretion gene clusters, which just complicates everything. But yeah, they find that both secretion systems, or like secretion system 1 is actually the most important in this case, to allow it to enter this niche of the gut. And if the Citrobacter rodentium doesn't have that type 6, then it just can't get into that niche. And that's probably because of, they trace it back to a microbiome member that exists inside of that space. So like if you change up the microbiota in the mice and you give them a microbiota that's more susceptible, then you could have Citrobacter rodentium that didn't have type 6 secretion systems get into that niche. So yeah, there's basically it's like this gene is important in the context of the microbiome and who's there and who's sort of like protecting, I guess, the guts of our mammalian friends, the mice.
Yeah, I mean, this is quite interesting. My supervisor used to study Citrobacter rodentium, and I remember like she changed the labs. So she moved from like a lab in the UK to a lab in New Zealand, and her Citrobacter rodentium model stopped working in New Zealand, and it was baffling as to why that stopped working. And I think this kind of indicates why, because different mouse colonies have slightly different microbiomes. So if the content of microbiomes, and they do this, they actually test from different like kind of mouse colonies that have grown up almost independently for a couple of years, and then checked what the differences between them, and mostly differences were between the microbiomes to see what makes them more or less susceptible. And they found that there is a difference, and that's kind of a really interesting thing, because this is being used as a model for human E. coli kind of responses. And so it's really interesting that they find that for mouse, having a certain members of the microbiome community present there or absent can change whether this Citrobacter rodentium can stick to the gut or not. And the question in my mind is, well, how does this relate to the human versions of this bacterium? Because it sounds like there is almost something interesting there that relates to humans.
Yeah, I mean, from the therapeutic mindset, one could imagine like the right mix of probiotics, right, that prevent certain bacteria from being able to colonize you, certain pathogenic bacteria from being able to colonize you. But from what I've heard in the space, it's like there's so many different. Well, first of all, just in this example, there are 2 different types of secretion systems. They may also have 2 sets of effector pools, so things they like to inject. This particular paper does focus, I think, on something. I think they have like one effector that they're looking at that, like, if it gets injected into like the native bacteria, it's able to kill off those bacteria and then I guess find their niche. But it gets complicated quickly the deeper you dive into this. Probably very unexpected ways, because we've said before, like, at this scale, like, there's horizontal gene transfer going on. Who's related to who? How did these modules get swapped around? There's a lot of complexity going on. But yeah, I think knowing more about how it works inside of a model is giving us the framework for then bringing into the more the human case, right? So like, this is in some ways not related to a human disease or something like that, right? It's just, this is mice getting diarrhea from something that gives mice diarrhea. But all the relationships that are being interrogated and the tools that are being developed are helping to, understand how better to create how better to develop therapeutics on the human side.
I think that. Yeah, I think that's pretty much all I was going to say about the paper. So.
And yeah, I think that's all I want to say as well. So on to our last paper then of the day.
And this one was this analogy was created by me, the RNA wrestling analogy, because so diversity of bacterial small RNAs drives competitive strategies for a mutual chaperone. So in this paper, we're kind of talking about bacterial signaling and how bacterial small RNAs are important, because bacterial small RNAs can sit on an area of the genome and stop it from being transcribed, which is a very important function for turning genes on and off, or making sure that, say, a transcribed RNA only acts at a specific time.
Yeah, and it's something that happens inside of like, it's also known inside of eukaryotic cells that this is like a strategy to have quick turn on and shut off of gene expression, because you can imagine, so for a refresher, if people are not so fresh on their molecular biology, but you have like transcription, where an RNA polymerase is looking at DNA, creating mRNAs. And then there's translation, where a ribosome picks up that mRNA and makes a protein. And typically we think of the protein as having the function. So we think of a scenario we can think about that that argues for the usefulness of like having like these types of RNA control mechanisms is, what if you have to make a bunch of protein for a specific scenario, but you don't have like the mRNAs sort of like stockpiled? Then you have to wait for the DNA polymerase to make all those RNAs, right? To sort of respond quickly to make more of those proteins. But if you had a lot of mRNAs sort of floating around, just like sort of waiting to be activated, then you could quickly just say, okay, now it's time to make protein. You're sort of closer in the number of steps it takes to get to the protein from some signal. So yeah, so we have a system like this, I think, of like sort of bounding up RNAs, and it's also known in bacteria that there's a system, and specifically it's regulated by the small RNAs that don't, I guess they don't make their own protein necessarily, but they can go and they can attach to other mRNAs, or they also just might aggregate mRNAs in certain ways, depending. There's like lots of different ways in which this regulation layer can exist. But yeah, it gives us like a quicker way of regulating protein expression.
Which is where the HFQ protein comes in, because HFQ is almost like a carrier. It's this ring-shaped protein that can carry these bacterial small RNAs around the cell, and it carries them because it kind of binds to these small RNAs. So there's got specific areas where RNAs can bind. And
yeah, so like a little sponge that has all the things, all these relevant small RNAs on it. But how to release those small RNAs, it's known that other small RNAs bind to HFQ and then release the other ones that are bound. So the authors of this paper are trying to understand like really some, I guess, in-depth biochemistry about like what binds what preferentially and what releases what preferentially.
Yeah, so the question is, like, okay, so does this protein just release RNAs when necessary, or is it more? And what they find is it tends to be more of a fight. What happens? You've got one RNA bound to like HFQ, another one can sneak in and then push the other RNA out. And in fact, that this is the majority of what happens. What happens on HFQ is almost like an RNA wrestling match where where HFQ is the ring, and these two RNAs have to occupy the same space and to fight it out to find out which one is going to win or not.
Yeah.
And this is so what they do is they test a number of different RNAs that have different binding HFQ. So there are different classes that some bind to different areas and some bind to the same areas, and seeing like which ones can kind of win and figure out what are the reasons for them winning. Because I think there's always there's been this idea that the RNAs can compete, but the question is what makes a good RNA compete against it. So could it be?
Yeah.
And I think they go into a little bit of, so if you read this paper, they go into a bit of the nitty gritty of like where it binds and how avidly it binds, so how tightly it binds to it, and also kind of and so they also find that sometimes you can get RNAs that bind to different areas of the protein, so they can both bind it at the same time, and also it depends on like the different parts, so they can truncate some of the proteins to change their binding properties, and so it's kind of interesting in the signaling perspective, because if you can have like two RNAs that are doing different functions binding to this protein, then the question is how does that affect ongoing signaling?
Yeah, so they don't really give us they give us a lot of like hypotheses on how it might feed back into like having cells be healthy and adaptable in many different scenarios. This paper is focusing in on those binding affinities, and it does so in a method that I feel like is becoming more and more common in these types of biochemistry interrogations, is like single molecule imaging, where we're not looking at stuff. We're not looking at stuff or I guess we can look on an average, but we see like events happening across like all these different molecules of HFQ bound to a microscope slide. And like that's similar to like when we looked at like remdesivir being like with the molecular tweezers or whatever, like that it's not the same technology, but it's in the same like theoretical, I guess, like approach to looking at biochemistry, where we know that in biology, like not all the molecules are going to do all the same thing when they get interacted. And actually, the proportion of molecules that do things is where biology works, in the fuzzy proportions, right? Like 50% of the molecules do this at this concentration, and then 80% of the molecules do this at this other concentration. And looking at single molecules on a slide helps us suss that out better than averaging it all together in some sort of reaction.
Yeah, I mean, I think it's a really interesting focus because things like this allow us to get a lot of data out, and also like because when you average things out, you tend to like chop off the bits of the outliers. But even though, but now this one kind of takes everything into account, because even those outliers can be very important. So, for instance, in like 8 to 13%, they found like 4 RNAs could bind to something, or like sometime like some crazy things could happen. And thing is, like, actions that happen in the body that only happen like 10% of the time still might happen a lot, simply because there are loads of reactions.
Yeah,
so
absolutely.
It's not something that we can necessarily ignore as being completely non-functional. So
and we also don't know like the sort of like we're studying these biochemistry in isolation, but inside of the context of the metabolism in a cell, there could be mechanisms to look at some of those, like to amplify the signal that's coming from those rare events, right? And so yeah, it's just like, I guess this is just where we are with sort of modern technology and science, is that like we have this crazy resolution onto what's happening at a molecule level. And you know, the challenge is always taking those observations and then translating them into the organism and sort of building up that complexity. But yeah, it's a really cool way that this paper contributes.
Yeah, and I think that's all I've got to really say about this paper.
Yeah, so yeah, I think that's all we have for this week as well. So join us next week for more cool microbiology news.
Yeah, we finally kept it to nearly just an hour, which is a real victory, I feel.
Sometimes we ramble on for an extra half an hour, right?
Oh, sure. So how's your holidays been? But you know, thank you very much for joining me, and thank you all for watching. Just pulling up my end title script as well, which is terrible because I cut and pasted I'm out of practice, so I actually cut and pasted the intro script twice. So I'm gonna wing it. Danny, lead me in.
So we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, just let us know in the comments. You can reach out to us over Twitter or Mastodon. And yeah, this is the peer review process where anyone can sort of chime in with their 2 cents about papers. So if you think you have something to add or found something unclear, just let us know.
Yeah, thank you very much for watching. Thanks for coming and nerding out about all things small. I've lost my script, so I'm just going to say it's been a pleasure chatting with you, Danny. It's always so much fun to catch up with this. Sorry we've been away for a month. I've had health issues, so but yeah, hopefully we'll be back soon.
Back in 2 weeks
with more 2 weeks with more cool microbiology stories. So goodbye for now.